Okay, so quick review. That when we work genetics problems, as you have successfully done a couple of times now, you know the rules. You pick a letter, uppercase is dominant, lowercase is recessive. You try to pick one that looks different, upper and lowercase. You make your Punnett square. You put one parent's alleles above, the other to the left. What you might not have known before this class is what those letters represent. I tried to emphasize this last week. I'm going to emphasize it again because it's about to become real important. Those are not the parents. That and that is not mom and dad. That and that is the sperm and eggs that mom and dad can potentially make. This is a probability square. And so what we're doing is we're saying dad, when he makes his gametes, when he makes his sex cells, they're either going to have the dominant or the recessive allele. And mom, when she makes her egg cells, they're either going to have the dominant or the recessive allele. Segregation has already happened by the time you make this Punnett square. And so what these represent are the possible sperm and eggs that can meet inside the square. The kids are the products of sperm meeting egg in the 4 different ways that can happen. Does everybody get that? Because it's about to become super important. Because the next thing Mendel starts to look at is, okay, we got hair color and eye color in people, or we have flower color in peas, you know, purple and white flower color, and we have plant height, tall and short. Well, do they affect each other? That was the question. Does it just because you get the big P for purple, is that going to influence whether you get the big P for height? Do they travel together somehow? That's his question. And what he discovers is, at least in his case, in the case of the traits that he picked, no, they do not. They do not influence each other. That's Mendel's law of independent assortment. Yes, that's the part you start writing. Now, the real truth is that as we get into more advanced genetics, we do find that sometimes they affect each other. And it depends on whether these genes are on the same chromosome or not, but that's later. For now, we can assume, no, they don't affect each other. They don't affect each other. Just because you get the big beef brown hair doesn't mean you get the big beef brown eyes. Which means it should be fairly easy to track 2 traits, 2 genes at once. And when I say fairly easy, I mean, okay, more complicated than we've been doing. Because if we're going to track 2 traits at once, what that means is there's now 4 possible sperm and eggs mom and dad can each make. Which means how big is our Punnett square? How big is our Punnett square if there's 4 sperm and 4 eggs mom and dad can each make? Armani? 4 what? Well, it was 4 squares before, wasn't it? 2 by 2? So what is it now, Chris? 4 by 4, or 16 possible kids. That's what we're about to deal with, just so you know what's coming. Okay, let's see if we can do this. I'm just going to let you kind of fight it out for a minute here. Grab that blank sheet of paper. You don't have to rewrite the problem. I just want to see what you can come up with Punnett square-wise for this problem. In humans, brown hair is dominant to blonde hair, and tall is dominant to short. What is the expected phenotypic ratio if two individuals mate who are heterozygous for both traits? Now, what that's going to look like, what can I do white? I want white. Anyway, what that's going to look like is, say this is dad, and this is mom. There's your two parents. Consider that paper, scratch paper for whatever you want to try. See if you can get a proper Punnett square set up, knowing what we just said. It's got to be 4 by 4, right? So what does a 4 by 4 Punnett square look like for those two parents? What's it look like? Making a good try, but making the same basic mistake. And the same basic mistake is this. Let's say this is dad. When he produces a sperm cell, does it have just the hair color gene on it? Shouldn't it have also the height gene on it? So if we know it's one of these. And we're saying dad makes, and this is the first sperm cell dad can make, and you all are just putting a big letter B there and no T. That's not right, is it? That's not a complete sperm. A complete sperm has to have one of each. So what you're really doing for each of these slots is saying, okay, how many different ways can dad put together a B and a T? So give me one. What's one thing, one combination of B and T that can come from this dude that can go here? Big B, big T is your first slot. Go from there. See what else, what other 3 combinations are there of a B and a T? By the way, keep the B before the T. Got a whole sheet of paper to work with. If it's not erasing, well, just make a new one. Doesn't take that long. Try to stay consistent with the B being before the T in your pairings. But yeah, most of you are getting it just fine. So let's look at this in kind of an organized way. So how we set this Punnett square up is we take these two parents and see how many ways they can put a B and a T together. So you said the first one. It's just a reminder that we're talking about sperm and eggs. Big B, big T. So we take it from here and here. There's your big B, big T. What's another way? What's the second one you chose? Abby?
Big B, little T.
Big B, little T. Yeah, he's got those, so he can do that. And that's different from the first example. Tristan, what's next? Take the 2 inside ones, little b, big T, and then, of course, the last one being both littles, right? The 2 little ones, those go there. So down the left side, what do? And she's got the same genotype as he does, so we do exactly the same thing. Big, 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 little, little, big, little, little. And now, the job is this. When you write those kids in, remember, these are the 16 possibilities of kids these parents can have when they flip that coin, or the 2 coins, or 3 coins, whatever, 4 coins. When you write them like we wrote the parents. In other words, they should all be B, B, T, T. Don't go B, T, B, T. That's it. Very hard to tell what the phenotypes are when you have letters split up like that. You want all to be B, B, T, T with the letters that the parents gave in that square. So go ahead and carefully fill out your Punnett square now. And if you are confident at this point, it's probably time to transfer this into your notes. Go ahead and draw this carefully and cleanly and clearly in your notes so you have a good example of what to do with the Punnett square like this, because you are going to need to come back to this, I think, as you do these problems that you're about to get. This is where you got to be real careful about your letters, right? This is where you become very glad that you didn't pick a letter like C or O or S, that it's hard to tell the difference. If you do pick a letter like that, you want to make it real exaggerated between lowercase and uppercase. Check your top row. Make sure it matches mine. You guys are quick, by the way. You got it more quickly than I'm used to, so that's good. Even on a Tuesday morning, half catatonic. Now, though, because we haven't answered the question yet. Remember the question? The question was, what is the phenotypic ratio in the offspring of this brown-eyed, no, was it hair and height? Yeah. Brown-haired, tall, heterozygous man and brown-haired, tall, heterozygous woman. What is the phenotypic ratio of the kids? Well, first thing you got to figure out is how many phenotypes are there? How many phenotypes can the kids even have? How many different ways to look are there? There's not 16 different ways to look, right? There's several different genotypes. But as far as how these kids look, there's only 2 hair colors and 2 heights, right? So a kid, what's one way a kid can look in this Punnett square? Dominant for both, right? He can show tall and he can show brown haired. So that's where we're going. We need to identify all the different ways a kid can look and count how many are in that Punnett square. This is the kind of the part where it's easy to screw up, especially if you can't read your letters. So you want to be clear. So this is the same square. I just don't have it color coded. Let's start. How about you just circle in that Punnett square, circle all the kids that are brown haired and tall. Put a circle around each of those children. Possible children, potential children. It's a bunch of them, right? Because all they need is one big B and one big T. As long as they have one big of each letter, doesn't matter what the other letter is. So you're counting those. If you laid it out like I did, it makes a fairly convenient pattern. How many did you get? How many brown-haired tall kids we looking at here? Abby? 9. And they make a nice little triangle. 1, 2, 3, 4. All the top row, all the left column, and then this one and this one, and we've got our 9. Right? Those are all the ones that have at least one big letter for both genes. Good so far? And keep in mind that whatever these numbers add up to, it better be 16. Right? Because there's 16 boxes in this square. So however many numbers we have, they better add up to 16. So 9 is our first number. The next way, let's look for a kid to look. Let's do brown hair but short. And let's put a, what did I do? Square? Let's put like a diamond shape around those so you know they're different. All the brown-haired but short. That means at least one big B, but two little T's. Find them. How many did you find? 3. Another triangle too, right? 1, right? 1, 2, and 3. Good. Let's put a square around the blonde tall ones. That is 2 little B's, but at least one big T. Should be getting easier as you knock out these blocks, right? You've only got a few left. How many of them are blonde but tall? 3. 1, 2, and 3. And there's one left, and that is our blonde-haired and short person. So I don't care what symbols you use. I don't care how you do this, but the most successful way I've seen students do problems like this is to just mark the different phenotypes differently until you get through the whole Punnett square, okay? But I will tell you this, when we're following 2 traits and the 2 parents are heterozygous for both, you're always going to get the same thing. You're always going to get the same ratio. And by the way, we'll almost never ask you for a genotypic ratio in this case, because if you did that, it'd be kind of a nightmare. There's an awful lot of different genotypes. We're probably just going to ask you for phenotypic. And in this kind of cross, the phenotypic ratio will be 9 to 3 to 3 to 1. 9 showing both dominant. 3 showing dominant for the first, recessive for the second. 3 showing the reverse, right? Recessive for the first, dominant for the second. And 1 out of 16 with a double recessive. Still need to remember that this isn't predicting the way 16 kids will turn out. If this couple has 16 kids, this is not telling us that 9 of them will be dominant. This is telling us every time this couple has kids, there is a 9 out of 16, that is a better than half chance that the kid will be dominant for both. And there's only a 1 out of 16 chance, very small chance, that the kid will be recessive for both. We good? So that cross, that particular kind of cross, 2 traits and heterozygous for both traits in both parents, has a name. It is called a dihybrid cross, and you should write that down. As soon as you see that, once you get to know this well enough, you won't even have to do the Punnett square. You'll just know, hey, it's 9 2 3 1. Nine to 3 to 3 to 1 would be a good number, a good ratio to memorize. But it only applies when both parents are heterozygous for 2 different traits. And that's as complicated as it gets. If you know how to do that, it's just a short step from there to start to change that Punnett square if somebody's not heterozygous. If somebody is homozygous for one of those traits, basically your Punnett square gets easier. So here's some practice with that. We're going to say side 1 is what you need. And side 1 is going to be due tomorrow, along with your guided reading. Just the front side, just 1 through 5. Come on. Huh? I'll get there. I'm just being weird about my packing here. So this is going to start almost exactly like this, right? The first problem is going to be very similar to the problem you just did, in that it's 2 heterozygotes. It'll throw a little bit of math in there, but it's easy math. And then it'll start messing with it a little bit and having you figure some stuff out. So please use this last 12 minutes to do at least that first and second problem. Use your own paper if you like, or if you feel like you have room on the paper itself, do that. That's fine. Remote folks, I'll make it a PDF and you can either do it in Kami, but I honestly, I guess, would probably recommend doing it on paper and then taking pictures. Show all Punnett squares for all 5 of these, please. Any questions? All right, get to work. Yeah. No, I need you to be able to interact with me.